Breaking the wall of ineffective CAR T cells. Anthea Wirges. Falling Walls Lab, Berlin-Adlershof.
Welcome everybody. B-non-Hodgkin lymphoma is the 7th most common malignancy, and worldwide more than 200,000 people succumb to it every year. It is a heterogeneous set of disease, and certain subtypes tend to a frequent relapse. The patients that are relapsed and refractory to the first-line treatment don't have a lot of options left, and their best chance at the moment is CAR T-cell therapy. Within CAR T-cell therapy, T-cells from the patient's own blood will be isolated, modified ex vivo to express a so-called CAR, the Chimeric Antigen Receptor. This is a fusion protein combining the antigen binding domain of an antibody with the signaling domain of a T-cell receptor. After expansion, those cells are reinfused back into the patient, where they specifically find and destroy the tumor cells. So far, we have 4 FDA-approved CAR T-cells for the treatment of relapsed and refractory B-NHL patients, but all targeting the CD19 antigen. And for all of those CAR T-cells, high relapse frequencies of 50% are reported, mainly due to loss or downregulation of the CD19 antigen. That's why new target antigens are needed to break the wall of ineffective CAR T-cells. We identified CXCR5 as a novel and a better target antigen for CAR T-cell therapy of B-NHL patients. CXCR5 is the B-cell homing receptor, guiding the B-cells and the malignant counterparts into the B-cell follicle of the secondary lymphoid organs. In contrast to the really broad expression of CD19, CXCR5 is uniformly high expressed on primary patient samples with B-NHL. But CXCR5 is also present on a small population of T-cells, the so-called T follicular helper cells. Those cells are also located inside the B-cell follicle, and they are part of the tumor microenvironment, so they are providing tumor cells with growth and survival signal. The unique mode of action of our CAR is now that it targets the tumor and the tumor microenvironment at the same time by using the same cell and the same target antigen. This was proven by us in a preclinical setting. We have an in vivo mouse model that demonstrates the really efficient anti-tumor activity of our CAR T-cells. Furthermore, in an in vitro setting where we co-cultured various patient samples together with different CAR T-cells, we could prove that only our anti-CXCR5 CAR is able to destroy these tumor-promoting population in the tumor microenvironment. And we are really lucky that in cooperation with the Charité, we will explore the safety and the primary efficacy of this approach in the clinics, hopefully in the beginning of next year. Thank you for your attention.